cell technology. We're obviously one of the most advanced companies in the cell therapy space, especially in the pluripotent cell therapy space. And so we're going to hear from Ted Myles, Executive Vice President of ACT.
I must say I am extremely green with balance sheet envy after watching the Sangamo presentation. It really is a roadmap for what we're trying to do at Advanced Cell Technology. Advanced Cell's been around for a long time. It has not accomplished all the great steps that Edward laid out on his plan, but we certainly have all those steps in front of us. And what we're doing now is we're in the transition mode. I'm actually acting as interim president in addition to being CFO. We're in the process now of really transitioning the company from a very, very heavy emphasis on research, which has generated a lot of value. Much of that value I feel we haven't unlocked yet. And we're really building up, now that we're deep into phase one and with a real look to phase two, we are building up a clinical organization, really shifting the emphasis of the company from research and purely research to development, and still funding the what we refer to as sort of the golden goose that's creating a lot of great technology in the research programs, but really focusing and shifting and allocating resources so that we can execute and succeed in the clinic, which we believe will certainly bring in more funding, dilutive and non-dilutive, which will fund more research. So I'll get into that as we go through the presentation. The Sangamo presentation was a terrific lead into really what we're hoping to accomplish over the next 12 to 18 months. So cautionary statements, I'll be making some forward-looking statements. Please read our risk factors and understand that what I say may differ from reality over time. So a couple quick investment highlights on Advanced Cell Technology. As I mentioned, it's really been heavily focused on research. Our company was doing embryonic stem cell research through the 2000s when nobody else was doing it. And what that's created is a real center of excellence and very high capabilities and world-class cell biology, and a really rich pipeline of clinical and preclinical assets. Additionally, it's given us a chance to create a very leading, what we believe to be a really valuable IP position. We don't think this has been fully unlocked yet, but we intend to do that in the near future. We are deep into phase one in 3 clinical trials, 2 for Stargardt's macular degeneration and 1 for age-related macular degeneration. I'll get into that in more detail shortly. The Stargardt's version of macular degeneration is an orphan indication. AMD, as many people know, is one of the leading causes of blindness. It's reaching epidemic status. Several hundred million people worldwide are projected to be afflicted with this disease and ultimately blindness by the year 2020. It's really a devastating impact on social healthcare costs as well. And as part of the rebuilding of the company, we've brought in a couple of very experienced management team members and also a very strong board of directors we're very proud of. So the pipeline, and much like what Edward touched on, our pipeline, we really are looking at this as defining our strategy. We view the macular degeneration franchise as being the primary asset in the company, and we're driving forward and resourcing the company appropriately so that we can execute and succeed in phase one and then build up to phase two, obviously. The other ophthalmology programs are really promising in the proof of concept phase. We see a lot of value there. Our recently hired EVP of Clinical Development is actually an ophthalmologist. We think stem cells have a good place in ocular indications, so there's starting to be a more narrow focus, not just on how we allocate to capabilities, but also in a disease area. Further down the line, we've had a lot of good activities and a lot of success in the mesenchymal stem cell area. Platelets was a program we talked about for a long time, made a lot of good advances. Government funding that really is required to move that thing to the next level hasn't materialized, so we've shelved the program until something does materialize. So talking a little bit about the retina, that's our primary focus in the company right now. The retina is at the back of the eye, and as you kind of build out the cross-section of the back of the eye, what you see is a number of layers here. So you've got the the nerve fiber, the ganglion, and the photoreceptors. Everyone knows the rods and cones. That's what transitions light into images. And then the RPE layer. What we're really focused on at Advanced Cell Technology now is this RPE layer. These are critical cells. It's a single layer of cells that line the back of the retina, and they are the life support system to the photoreceptors. As they go away, photoreceptors and therefore vision starts to go away. In macular degeneration, or age-related macular degeneration, it's thought to be the constant accumulation of UV light that damages this RPE layer over time. You get to be 67 years old, RPE layer fails, and the life support system for the photoreceptors fails. With the failure of RPE layer, you've got degenerative diseases in the macula. I mentioned Stargardt's disease. This is a early onset. It's a genetic disposition that creates the failure of the RPE layer. You could have a patient in their 20s that starts to get this kind of blur in the center of their vision, and over time it'll grow out and cause total blindness. About a 10 or 15 year track, so you can imagine how devastating that is for someone in their late teens, 20s. Age-related macular degeneration, it could start it starts to increase in prevalence as you get into the 40s and 50s, but really in the 60s and 70s we start to see a lot more prevalence of age-related macular degeneration, and again, 10 or 15 year slow, steady progression into blindness. Of course, myopic macular degeneration we're talking about now because we're about to launch a phase 1 trial there. That's the failure of the RPE layer due to the elongation of the eyeball. So our therapy is the same cells. We've coaxed human embryonic stem cells to become RPE. And what we're doing is injecting into the back of the eye, and what we're seeing is these cells are grafting. When you think about a golf course with divots, we're injecting new grass, if you will, and in many cases it's sticking and it's growing to confluence and replacing that lost RPE layer. So one of the reasons we're very excited about this program is there's a lot of attributes that make this very appealing to a big pharma company, but we're in the enviable position of having good attributes that this is doable by a smaller company. Once we fund appropriately, we can execute on this without a partner. There's a small dosage size, 200,000 in our trials, 200,000 cells per dose. We have the capability now to make 100,000 doses a year in our facility. So scale is not an issue in the near and intermediate term. The eye is an immune-privileged site, so you don't have some of the other issues that stem cell therapies would have in the spine or some of the other areas. The eye is largely immune-privileged. We use immune suppression in our early trials in an abundance of caution, but it's taken a lot of the complication out of the therapy. It's ease of administration. We're not reinventing surgery. There's no special devices required. It's a vitrectomy with a little bit of special technique that our KOLs have been able to develop and pick up fairly quickly, and they think that the rollout and the training of this could be very successful. In the eye, you can measure and observe what's happening almost at the cellular level, so it gives you a chance to really see what's going on, and it's been tremendous information for us as we've treated 34 patients. We're taking that learning and it's helping to inform our phase 2 strategy. And as I mentioned before, it is an absolutely enormous unmet medical need. So we think it's got all the good aspects that are worthy of further investment. I mentioned the prevalence. They start to increase almost exponentially with every decade of age after 40. And then at the same time, we all know that the population is aging and people are living longer. So an otherwise healthy 75-year-old may have 15, 20 years of good, high-quality life ahead of them. And blindness obviously puts a big damper on that. The Lancet just came up with an article a couple of months ago, 300 million people by 2040. It was a very, very big issue. I touched briefly on our GMP process. This is something we've invested in early, and we're very proud of what we've achieved here. We believe we can serve the market, certainly in the U.S. for the intermediate term, in our facility in Marlborough, Mass. We'd have to hire a couple more technicians to scale up beyond that, but we've got the manufacturing pretty locked down. We've invested in a GMP suite, so that's not going to be a rate limiter as we get into later stage trials. The design of our study, initially, before the FDA would allow us to start injecting human embryonic stem cells into eyes, you can imagine the pushback and the risk aversion to this several years ago. We were only allowed to inject into very, very, as one of our physicians said, very dead retinas, really, really advanced stage patients. There was a mountain of preclinical data that we've done to ensure that these cells are fully differentiated and aren't going to turn into something else, or teratoma or anything. And so we endeavored on that first cohort and 50,000 cells into a number of patients, saw good safety, continued with 100,000. And then in early 13, we got approval to expand into better vision patients. We thought, we really view this as a nod from the FDA that, okay, safety is there. We can risk a, call it not so dead retina or advanced stage patients, but still there's some vision to lose there. What we do is we treat the worst of the two eyes and leave the fellow eye or the other eye as a control. So we're progressing through this trial design now. We've got the dry AMD study and the SMD study going on in the US. And in the UK, we've got the SMD study. And we're just about to start. I hope to be able to announce it soon, the initiation of the MMD trial. Again, that's a expansion of our RP franchise, same cells, same basic affliction, only the cause of the disease is slightly different. Really fortunate, one of our top assets as a company is the company we keep, and we've got some of the world leader institutions for eye care involved in our trials. Tremendous thought partnership for a small company to have such great sponsorship in our trials is really beneficial. I'll be meeting with most of these folks in a couple of weeks at Arvo, and they are highly engaged. We get a lot of thinking from them. We're all focused on how we end phase 1 and get on to phase 2. I mentioned the surgery. I've personally never done a vitrectomy. I hear it's not particularly hard. There's a lot of them done, more than 1 million per year. It's a big piece of a lot of these folks' practices. And our therapy, delivery of our therapy is basically a vitrectomy. The surgeons that are involved in the trial now are very excited about scalability, the fairly easy use of administration here. And they don't think it's going to be a rate limiter at all. It'll be pretty easy to train up. As we go from KOL to a broader set of physicians, we think we'll be able to train and scale fairly quickly. A couple of years ago, The Lancet published the data on our first 2 patients, and what we saw, and what we continue to see, no adverse effects. Oops. Someone needs to register their software. Oh.
The red dots at the top of that box.
I'm a PC guy, not a Mac guy, so this is going to find me out there. Thanks. That red dot, thanks. And what we're seeing is, as we're studying these patients and the images, we're seeing that the cells that go in are, as I mentioned, the new cells are finding this divot, and then they're replicating. It's replication to confluence. They're replicating until they feel cells like them next to them, and then they stop. So they're really kind of filling in the divots. It's pretty amazing. We're seeing good engraftment. And over, some of these patients have been treated for more than 2 years. The cells are still there. There's persistence. In some cases, there's been some visual acuity signals, and those are persisting as well. The key for phase 1, of course, is safety, and all of our docs feel very comfortable with the safety that we have experienced, which to us is obviously very important. Here's the phase. We haven't released interim top-line data yet. We're not going to. We're hoping to be published in a journal in the near future. But I can tell you, 34 patients treated, very clean safety profile, and as we've discussed in the past, some strong indications of efficacy in some cases. I talked about the initiation of phase 1. Myopic macular degeneration, again, from us to the investor community, we feel this is a strong signal and endorsement of what we're seeing in the AMD and SMD trials. We're investing further in the program. It's the same cells, same surgeons, largely the same patients, just they have this issue for a different reason. The eyeball extends and that cracks the RP layer. We we feel that putting our RP cells back in could help help these patients. Very big issue. Seventh leading cause of blindness in the United States, and it's a very high cause of blindness in Asian populations. Talk about our intellectual property. We've got dozens of issued patents, several hundred pending, and we feel, because we were really the first ones in this space, we've gone out and gotten some very good, strong claims issued and pending that we feel will protect us as the rest of the field catches up to advanced cell technology. We feel we'll be in really good shape in terms of patent protection. So moving from the RPE program, these other ocular programs we're seeing, I'm not going to talk for too long about them. It's preclinical, it's early, but it's an example of the stem cell expertise and this research component of the technology creating other opportunities behind the RPE franchise. There are a number of other cells that we're targeting within the eye, cornea, applying mesenchymal stem cells into the eye. The retinal neural progenitor cells could create new photoreceptors. There's a lot of opportunity here, and we're continuing to invest carefully in the preclinical programs, advance them to a certain point, and then make further investment decisions. So we talk about photoreceptor replacement. This is one of our more exciting early programs. So outside of ocular, the mesenchymal stem cell program, we recently had a paper published about this. We feel that our mesenchymal stem cells are more potent. They're human embryonic stem cell derived MSCs. They're more potent, and we can also manufacture them in more abundance, in higher abundance. They're involved in immune regulation, autoimmune diseases, where it's immune response to yourself versus an allergic reaction, immune response to a external factor. Very early studies here. The intent is to move them into proof of concept and then partner them off. Really tearing the sheet out of Sangamo's business plan, where the core asset being in the ocular space, and some of this very valuable, but just not critical for us to move forward on. ACT is not going to look to go head-to-head with Biogen on an MS trial, but we feel that some of the companies that are involved in MS could reap real benefits from our MSC cells. So, as I mentioned, large supply of very potent MSCs. So, very experienced, dedicated management team. I'm filling in as interim president. In the meantime, we're open. The search is open for a full-time CEO. We look forward to that addition. Dr. Robert Lanza is our CSO, has been for a long time. Interestingly, he was just named one of Time Magazine's top 100 most influential people. So he gets to go to a fancy dinner in New York, and I think it's really pretty exciting. Dr. Eddie Angeli joined our company in January. Very experienced ophthalmologist, has run multinational, 1,000-patient plus clinical trials across the globe in the ocular space. Very successful clinical development, regulatory guy. We're fortunate to have him, and now he's taking our team and professionalizing the group and building up the capability in the company so that we can have the highest degree of success at the end of phase 1 and into phase 2. And as I mentioned, the board of directors has been overhauled in the past couple of years as we're sort of growing up and shifting from the legacy of the company into building a world-class biotech. Having Mike Heffernan and Bob Langer and Greg Perry on the board has really helped elevate the way the company performs. So thank you for your time. I think I have a minute or two for questions.
Thanks very much.
Any questions for Ted? I mean, I'll ask the question that you probably get quite a lot and you're probably sick of, but ES versus IPS, and any thoughts about moving in that direction?
So we think IPS is a long way away from clinical development, and I get asked a lot about, about investment in the platform versus investment in the therapeutic asset, and we want to do both.
Yeah.
We think the fastest path to additional funding is clinical development success, which will enable more research, and therefore we'll start investing in platform. But for now, our bet is human embryonic stem cells, and we think IPS is exciting, but many, many years away.
Okay. Thanks very much.
That's great.